Hello everyone. So now that we all know what proteins are, what are the different levels of structures, in this chapter we'll be talking about a particular class of proteins called enzymes. As you can see from the objectives, this is a pretty content heavy chapter in the sense we have lots of things to talk about here, and as a result of which this is also a content heavy chapter on your exam. So what are enzymes? Enzymes are simply proteins that act as catalysts within cells. Catalysts increase the rate at which chemical reactions occur without getting consumed or getting altered themselves. As a catalyst, an enzyme can facilitate the same chemical reaction over and over again. In doing so, an enzyme is not consumed or altered. Enzymes are highly specific in the types of reactions they catalyze. This specificity of the enzyme comes in part from their specificity to their substrate. A substrate is a reactant or a substance upon which the enzyme acts to initiate the reaction. It is important to note that all enzymes are proteins, but not all proteins are enzymes. Enzymes are a subcategory within proteins, some of the other categories being transport proteins, structural proteins, signaling proteins, and so on. So enzymes have a particular nomenclature. They have a recommended name and they have a systematic name. The systematic name is designated by the International Union of Biochemistry and Molecular Biology, and a lot of times the systematic name is very technical. So for the most part, most enzymes are known with their recommended names. And it is easier to distinguish an enzyme from one that's not from a protein that's not an enzyme, because for the most part enzymes end with the suffix ase. You have urease, you have sucrase, you have lactate dehydrogenase, and so on. Now there can be some exceptions, like in the case of trypsin. Okay, so not every enzyme ends with ase, but for the most part, a lot of enzymes do end with ase. Additionally, the prefix that comes before the ase tells you about the substrate. Okay, so this is a urease, which means that this particular enzyme uses urea as a substrate. Sucrase, this particular enzyme uses sucrose as a substrate. This enzyme uses lactate dehydrogenase as a substrate. Now to classify the enzymes, there are 6 broad categories that enzymes can be categorized in. Number 1 being oxidoreductase. Now these are a class of enzymes that catalyze oxidation reduction reactions. In other words, these enzymes facilitate the loss and gain of electrons between the participants of the reaction. Next, you have transferase. This class of enzymes catalyze the transfer of functional groups from a donor molecule to an acceptor molecule. Then you have hydrolases. As the name would suggest, this is a class of enzymes that catalyze hydrolysis, which means the breaking of a bond by the addition of water. Now the next two, or rather lyase and ligase, are the ones that get confused the most, so it's important to distinguish between them. We'll come to isomerase later. Lyase is a class of enzyme that catalyzes the breaking of chemical bonds by means other than by adding water. More often, the end products resulting from reactions involving lyase often have double bonds. Now, ligases, on the other hand, are a class of enzymes that facilitate the joining of two molecules to form one bigger molecule. Though they sound very similar, lyase and ligase, they practically have the exact opposite functions. One of them breaks, the other one joins. And lastly, we have isomerases. Isomers are entities that have the same chemical formula but different structural orientation in 3 dimensions. Isomerases are a class of enzymes that facilitate intramolecular rearrangements in which the bonds are broken and formed. Now let's take a look at an example of each one of these classes of enzymes. Starting with oxidoreductase, you have a lactate dehydrogenase. Okay, it is the name of the enzyme which tells you that lactate is the substrate. Dehydrogenase is the action of the enzyme, which means that it is taking off hydrogen atoms of a particular substrate. So what it's really doing here is lactate dehydrogenase is acting upon lactate as its substrate. It's removing the two hydrogen atoms and converting lactate into pyruvate. Now on this slide you will see a lot of other different coenzymes such as NAD, THF and so on that you really don't have to worry about at this point of time. You will of course be studying these later in much greater detail, but all you need to focus on right now is what exactly is the mechanism of action of that particular class of enzyme. So in the next class of enzymes we have transferases. Transferases catalyze the transfer of functional groups. So in this case you have serine hydroxymethyl transferase. So again, serine is the name of the substrate. Hydroxymethyl transferase. What it really tells you is that this particular enzyme is going to transfer a methyl group that's attached to a hydroxyl group onto another substrate. So here you have serine hydroxymethyl transferase acting on serine. It's got this methyl group that's attached to hydroxy group. It's taking that off, putting it onto tetrahydrofolate, and in the process it's converting serine into another amino acid, glycine. So that's how amino acids can get interconverted into each other. Hydrolases are enzymes that catalyze cleavage of bonds by addition of water. So here you have urease. The substrate is urea. Water is added to urea to decompose that into carbon dioxide and ammonia, and this is exactly how urea is excreted through the body in the form of carbon dioxide and ammonia. It has to be decomposed into both of these compounds. Next is lyases. Lyases catalyze cleavage of certain bonds without the addition of water, without hydrolysis. So here you have pyruvate decarboxylase. Again, pyruvate is the name of the substrate. Decarboxylase tells you something about the mechanism of action of the enzyme. It's going to decarboxylase, which means take carbon dioxide off of a particular substrate. So pyruvate decarboxylase acts on pyruvate, takes off carbon dioxide, in the process converts pyruvate into acetaldehyde. Then you have isomerases that catalyze isomerization of compounds. So here you have methylmalonyl coenzyme A. An enzyme called methylmalonyl coenzyme mutase just rearranges some of the atoms on this particular substrate, converts that into succinyl coenzyme A. Now what it's really done here is the methyl group which was branched out, it's turned that into a linear methyl group. In the process, it's converted that into succinyl coenzyme A. And lastly, you have ligases, which catalyze formation of bonds. Essentially, they join two molecules. So here you have pyruvate carboxylase, which with the help of ATP as an energy donor is attaching the carbon dioxide onto the substrate pyruvate. So in the process, it's converting pyruvate by attaching the carbon dioxide on the other side. It's converting pyruvate into oxaloacetate. Right, so what is an active site? An active site is something that's really important in the context of the structure of enzyme. On the surface of each enzyme is a special region called the active site, which provides a place where the enzyme and substrate can meet and interact. It's much like a lock and key. The enzyme's active site will only accommodate certain reagents, and there is only one type of chemical reaction that can be catalyzed by any given enzyme. So in that sense, they are extremely unique in the sense that they can only carry out a specialized reaction. The active sites contain amino acid sequence that creates a 3D surface complementary to the substrate. That's how the substrate can recognize the active site. The active site binds the substrate molecule to form the enzyme substrate complex. The enzyme substrate complex is converted to an enzyme product complex, and subsequently the enzyme and the product dissociate so that the enzyme is free to go and act on another substrate. Enzyme catalyzed reactions are highly efficient, proceeding from 10 to the power 3 to 10 to the power 8 times faster than uncatalyzed reactions. Each enzyme molecule is capable of converting 100 to 1000 substrate molecules into products per second. And this is the catalytic efficiency of an enzyme, which is measured in terms of what is called the turnover number of enzyme that we'll talk about a little later. So what role do enzymes exactly play in catalyzing a reaction? In order to understand that, first we need to understand what is the energy barrier. One of the indisputable laws of nature is that energy is required to bring a change. In this context, energy is required to convert the reactants into the products. In order to do that, you must have to cross the free energy of activation. The energy of activation is like a hill between the reactants and the products, and the reactant must be pushed over the hill in order to be converted into the product, in order for the reaction to continue. For every chemical reaction, a specific amount of energy, which is the energy of activation, must be applied to the reagents before the reaction will proceed. So what enzymes do is they increase the rate of reaction by providing an alternate pathway with a lower free energy of activation. Enzymes do not change the free energies of the reactants and products. Note here that the delta G, which is the Gibbs free energy, which is the energy difference between the reactant and the product, remains the same with or without the enzyme. What the enzyme is really doing is it's providing an alternate path with a lower free energy of activation, thereby helping the reaction proceed in a much more faster manner. So enzymes, again, note that while they lower the free energy of activation, there is no difference in the free energy of the overall reaction. Now, as an example here, we have hydrogen peroxide getting decomposed into water and oxygen. Now, the free energy of activation, which is the hill that you see here, has an energy of 75.2 kilojoules per mole. So, in the absence of an enzyme, let's say that you have one molecule of hydrogen peroxide getting decomposed with this free energy of activation. In the presence of an enzyme, platinum, the free energy of activation has now been reduced to 48.9. In the process now you have 2.7 times 10 to the power 4 molecules getting decomposed. In the presence of another enzyme called catalase, which even lowers the free energy of activation further down to 23, you have 6.5 times 10 to the power 6 molecules of hydrogen peroxide getting decomposed. So that is how enzymes facilitate reactions by lowering the free energy of activation and accelerating the reaction. Let's take a look at some of the enzyme terminology. Cofactors. Cofactors and coenzymes are sort of helper molecules that help the enzyme carry out the reaction. Enzymes can be kind of useless without these cofactors and coenzymes. A slight difference between the cofactors and coenzymes, though essentially they are both helping the enzyme, is that cofactors are inorganic metal ions, in the sense that the term inorganic here means without carbon. On the other hand, you have coenzymes, which are organic molecules with carbon. Many of these coenzymes are actually derived from vitamins, and that will undermine what importance vitamins have in our life when we talk about vitamins in the next chapter. But just as an example, you have TPP, which is thymine pyrophosphate, which is a coenzyme that is derived from vitamin B1. You have NAD, which is nicotinamide adenine dinucleotide, another coenzyme derived from vitamin B3 that is involved in oxidation reduction reactions. Prosthetic group can be either organic or inorganic, a metal ion. What sets prosthetic group apart is that these are cofactors or coenzymes that bind tightly to proteins or enzymes in our case. As if holding on for dear life, they cannot be easily removed from the enzyme. They are most often attached by a covalent bond. These are simply some terms. A holoenzyme is an enzyme with its cofactor. Apoenzyme, an enzyme without its cofactor. Zymogen. Zymogen is an inactive precursor of enzymes. A lot of times, a lot of enzymes are synthesized in their precursor form, in the sense that an enzyme is not synthesized as an active enzyme because you want that particular enzyme to be activated under particular circumstances. And that is why you have enzymes in the form of zymogens. These zymogens then can be activated by some peptide cleavage. A part of the enzyme can be cut off and that enzyme can be activated, or you can have an enzyme getting phosphorylated, which we will talk about later, and that phosphorylation can activate the enzyme. Then you have the turnover number, which defines the efficiency of a particular enzyme. It's simply defined as the number of substrate molecules converted into product by one molecule of enzyme active site per unit time when the enzyme is fully saturated with substrate. So the turnover number for lysozyme is 0.5 per second. The turnover number for catalase is 4 times 10 to the power 7 per second, which essentially tells you that catalase is a much more powerful enzyme capable of working on these many substrate molecules per active site per unit time as compared to lysozyme. Isozymes or isoenzymes are enzymes that differ in amino acid sequence and yet catalyze the same chemical reaction. Usually these enzymes have slightly different kinetic parameters such as binding affinity or different regulatory properties. So in other words, these are variants of an enzyme with some slight mutation in amino acid residues in non-critical area of the protein. Isozymes catalyze the same chemical reaction. An example here is lactate dehydrogenase. Lactate dehydrogenase has 4 subunits that can combine in different forms to produce isozymes. Lactate dehydrogenase catalyzes the conversion of pyruvate to lactate and back to pyruvate, but more on that later when you get to glycolysis and TCA cycle. Lactate dehydrogenase is composed of 4 subunits. The 2 most common subunits are LDH-M and LDH-H. Now these 2 subunits can form 5 possible tetramers. You can have a 4H, you can have a 4M, you can have 3 M's, 1 H, 2 M's, 2 H, and 1 M and 3 H. These 5 isoforms are enzymatically similar but show different tissue distribution. For example, the 4H isomer is primarily found in heart and red blood cells. The 4M isomer is primarily found in muscle and liver. Then you have the 2H2M form that is found in lungs. Then you have the 1H3M form found in kidneys, and then you have the 3H1M form that is found in the reticuloendothelial system. Now you don't really need to remember which form is found in which particular system, but all you need to get here is the fact that you can have isozymes which are slightly different proteins carrying out the same catalytic function. Now let's take a look at some of the factors affecting reaction velocity. The reaction velocity primarily depends on three parameters. One is the substrate concentration, second is the temperature, and third is the pH. Let's take a look at the substrate concentration and how it affects the velocity first. So what is the reaction velocity? On the y-axis here you have the reaction velocity, which is nothing but the number of substrate molecules converted to product per unit time. Now what's really happening here? So on the x-axis you have the substrate concentration. On the y-axis you have the reaction velocity. So what's happening here is the rate of an enzyme catalyzed reaction is increasing with substrate concentration until it reaches a maximum velocity. The leveling off of the reaction at high substrate concentration reflects the saturation of the enzyme with the substrate at all available binding sites on the enzyme. A significant portion of enzymes function in a manner that their properties can be studied using what is called the Michaelis-Menten kinetics. These enzymes follow a hyperbolic curve. Hyperbole is nothing but exaggeration. So what is happening here is at a very small substrate concentration, you can see an exaggerated response in the reaction velocity. The reaction velocity is taking off like a Ferrari. Okay, so at a very small substrate concentration, you have the reaction velocity taking off really fast, which is a hyperbolic response, and that is why these enzymes are called, they follow the hyperbolic curve and can be studied using Michaelis-Menten kinetics. On the other hand, you have another class of enzymes called allosteric enzymes that follow the sigmoid curve. It's called the sigmoid curve because it simply looks somewhat like S. So what happens in these allosteric enzymes is they are a little slow to take off. You'll see that the reaction velocity starts off very slowly, and then at a particular point, after you've had enough substrate bind the enzyme, which is called cooperative binding, it really sort of takes off after that. So these are called cooperative enzymes, wherein they need to have some form of substrate bound to the enzyme. What that does, it increases the affinity of the other enzyme active sites to the substrate. So once you have some substrate bound to the enzyme, it increases the affinity. It's called cooperative binding in the sense that it makes it easier for the other substrate molecules to bind to the enzyme. Next, you have temperature. Like most chemical reactions, the rate of an enzyme catalyzed reaction increases as the temperature is increased. A 10 degrees centigrade rise in temperature will increase the activity of most enzymes by 50 to 100%. In the case of enzymatic reactions, this is complicated by the fact that many enzymes are adversely affected by high temperatures. So as shown in this figure here, the reaction rate increases with temperature to a maximum level and then abruptly declines with further increase in temperature. So what's really happening here is it's inactivating the enzyme because the protein is getting denatured. So because the protein's function is dependent on its structure, as soon as the protein starts getting denatured, the enzyme loses its functional capability. The other factor affecting enzymes is the pH. So enzymes have a favorable pH range, which is their optimum pH. Extremely high or low pH values generally result in complete loss of activity for most enzymes. So it again all goes back to the ionization of side chains. You have particular side chains being ionized at a particular pH. The ionization state changes as the pH changes. Once the ionization state changes, it changes the structure of protein, which in this context changes the structure of catalytically active site, and the enzyme then just stops functioning. So this is the Michaelis-Menten equation. Okay, what the Michaelis-Menten equation really describes is how does the reaction velocity vary with the substrate concentration? Now, at low concentration of substrate, there is a steep increase in the rate of reaction with increasing substrate concentration. So what is really happening here is the catalytic site of the enzyme is empty, waiting for the substrate to bind, and at this point, the rate at which the product can be formed is limited by the concentration of substrate which is available. Now, as the concentration of the substrate starts increasing, the enzyme starts getting saturated with the substrate. As soon as the catalytic site gets empty, more substrate is available to bind and undergo reaction. The rate of formation of product now depends on the activity of the enzyme itself, and adding more substrate at this point is not going to increase the rate of the reaction to any significant effect. The plateau occurs because the enzyme is saturated, meaning that all available enzyme molecules are already tied up processing the substrate molecules. Any additional substrate will simply have to wait around until another enzyme becomes available. So the rate of reaction is limited by the concentration of enzyme. The maximum rate of reaction is characteristic of a particular enzyme at a particular concentration and is known as the maximum velocity, or Vmax. The substrate concentration that gives you a rate that is halfway to Vmax is called the Km, the Michaelis constant, and is a useful measure of how quickly the reaction rate increases with substrate concentration. Km is also a measure of an enzyme's affinity for, or in other words, its tendency to bind to its substrate. A lower Km, which in this case is indicated by the blue line, corresponds to a higher affinity of the enzyme to the substrate. The higher Km with the red line corresponds to a lower affinity of the enzyme to the substrate. So in this case, you have these two enzymes which may attain the same Vmax, or let's say they do attain the same Vmax. However, enzyme 1 has a much stronger affinity for its substrate, because of which it reaches its half Vmax at a much lower substrate concentration. The enzyme 2, even though it is as efficient as enzyme 1 in terms of reaching a Vmax that is the same as enzyme 1, because of its lower affinity to its substrate, its half Vmax is at a much higher substrate concentration. So this is essentially what the Michaelis-Menten kinetics is all about. Now, for those of you who are curious about equations and math, this is the Michaelis-Menten equation. You don't really need to memorize this equation. All it tells you is how the reaction velocity varies with the substrate concentration, which is given by V0, which is the reaction velocity at any given substrate concentration, is equal to the Vmax times the substrate concentration divided by Km, which is the Michaelis constant, plus the substrate concentration. So, some of the highlights of Michaelis-Menten kinetics that we already talked about, but it's always good to reiterate, is the Km. Km is the Michaelis constant. Smaller the Km means higher the affinity of the enzyme to the substrate. Larger Km, lower the affinity of the enzyme to the substrate. Now bear in mind not to confuse the Km with the Ka that we've earlier talked about. Ka is the dissociation constant of an acid, and in that case the large dissociation constant means a stronger acid. In this case, a large Km means a weaker affinity, or a not much weaker affinity of the enzyme to the substrate. Now that brings us to the order of reaction. Now, if you notice this hyperbolic curve, you'll see that in the initial part where the substrate concentration is much lower than Km, it's almost a linear line in this part. So if the substrate concentration is much less than the Km, the Michaelis constant, the reaction is said to be following a first order reaction, which means the rate of reaction is almost linear with the substrate concentration. On the other hand, if you have a substrate concentration much higher than Km, you will see that the reaction is proceeding at its own will. The substrate concentration is not really affecting the velocity of the reaction, which means the reaction velocity at a higher substrate concentration is independent of the substrate concentration and is said to be zero order. One of the assumptions of the Michaelis-Menten equation is that the substrate concentration is much higher than the enzyme concentration. In other words, your reaction will never run out of the substrate. Therefore, the limiting factor for maximum velocity in Michaelis-Menten equation is always the efficiency of the enzyme. Therefore, if you have the amount of enzyme, you will invariably have the V0 and Vmax at any given substrate concentration. Now, as good as the Michaelis-Menten equation or kinetics is in order to characterize the efficiency of the enzyme, you will notice that it is a curve, it is a hyperbolic curve. So that makes it really challenging to interpret when exactly the Vmax would be, or what exactly would have the Vmax be, which would affect the Km. So in order to get over this limitation of dealing with a curve, a couple of other guys called Lineweaver-Burk came up with an idea that could flip over the Michaelis-Menten equation and convert it into a line plot, which is much easier to read and interpret. Now, you don't really need to focus on this equation unless you are curious as to how the Michaelis-Menten equation gets converted into the Lineweaver-Burk plot. This derivation has nothing to do with, you won't be tested on this, okay? It is just in case you are curious to know how the Michaelis-Menten equation gets flipped over into a line plot. Please stay with me for the next couple of minutes, else you can just scroll forward. So this is the Michaelis-Menten equation. V0 is equal to Vmax times the substrate concentration divided by the Michaelis constant plus the substrate concentration. So if you take a reciprocal on both sides, what you end up getting is this equation here. This equation, the right hand side, can be rearranged as Km divided by Vmax times substrate concentration plus substrate concentration divided by Vmax times substrate concentration. So if you rewrite this equation, it becomes 1 over V0 is equal to Km divided by Vmax times 1 over substrate concentration plus the S and S are going to cancel out, so you have 1 over Vmax. Let's call this our equation 1. Now if you notice, this equation is very similar to the line equation, y is equal to mx plus c, where m, your slope in this equation, is Km divided by Vmax. Now how do you find a y-intercept in a line equation? All you do is substitute x is equal to 0, so that's what we'll do in equation 1. So if you substitute x is equal to 0, which is the substrate concentration, since that is our x-axis, is equal to 0, what we get is 1 over V0 is equal to Km over Vmax times 0 plus 1 over Vmax. Therefore, 1 over V0 is equal to 1 over Vmax. In other words, your y-intercept is 1 over Vmax. Now similarly, let's find the x-intercept by substituting y is equal to 0 in equation 1. So your velocity is your y-axis, so you put 1 over V0 is equal to 0 equals the right hand side of the equation. You rearrange the right hand side of the equation, and what you get is negative Km divided by substrate concentration is equal to 1, or in other words, your substrate concentration is equal to negative 1 over Km, or your x-intercept is negative 1 over Km. So your y-intercept is 1 over Vmax, your x-intercept is negative 1 over Km. Now with that, let's move forward to the Lineweaver-Burk plot. So we've converted the Michaelis-Menten equation into the Lineweaver-Burk plot because one of the limitations of Michaelis-Menten is it's not easy to determine the exact Vmax. So you take the double reciprocal plot where you convert the velocity into 1 over velocity, your substrate concentration into 1 over substrate concentration, and eventually your hyperbolic curve now becomes a line. This line is crossing the y-axis or has a y-intercept of 1 over v max, and it has the x-intercept at negative 1 over km. Now, let's take a look at what are some of the factors that can inhibit the enzyme activity. Any substance that can diminish the velocity of an enzyme catalyzed reaction is called an inhibitor. Now, there are two kinds of inhibitors. You have irreversible inhibitors and you have reversible inhibitors. Irreversible inhibitors covalently bind to an enzyme, thereby causing chemical changes to the active sites of the enzymes and cannot be reversed. A main role of irreversible inhibitors include modifying key amino acid residues needed for enzymatic activity. An example of irreversible inhibitor as a drug is penicillin, the one with the beta-lactam ring, which binds to an enzyme called peptidase, which is the bacterial enzyme involved in bacterial cell wall synthesis. And then you have the second class of inhibitors, which is the reversible inhibitors. Reversible inhibitors bind non-covalently to enzymes, and many different types of inhibition can occur depending on what the inhibitors bind to. The non-covalent interactions between the inhibitors and enzymes include hydrogen bonds, hydrophobic interactions, and ionic bonds. Many of these weak bonds combine to produce strong and specific binding. In contrast to substrates and irreversible inhibitors, reversible inhibitors generally do not undergo chemical reactions when bound to the enzyme and can be easily removed by dilution. Now, within reversible inhibitors, you have two important categories, which are the competitive inhibitors and non-competitive inhibitors. Competitive inhibitors, as the name suggests, competes with the substrates to bind to the enzyme at the active site. This type of inhibitor has an affinity for the active site of an enzyme where the substrate binds as well. The competitive inhibition can be overcome by increasing the concentration of the substrate, thereby outcompeting the inhibitor. Competitive inhibitors are often structurally similar to the real substrate. Non-competitive inhibitor, on the other hand, binds to the enzyme on a site other than the active site. So it's not really in competition with the substrate. So what it does is, you can have this substrate bound to the enzyme, but if the non-competitive inhibitor were to be bound to the enzyme that is bound to the substrate, it would still inhibit the reaction from proceeding, because when the non-competitive inhibitor binds to the enzyme to a site other than the active site, what it really does is the enzyme undergoes some structural modifications because of the non-competitive inhibitor being bound, as a result of which it's no longer able to facilitate the reaction. So, you have competitive inhibitor that binds reversibly to the same site, same active site that the substrate would occupy. This means that increasing the concentration of the substrate will decrease the chance of inhibitor binding to the enzyme. Therefore, if the substrate concentration is high enough, the enzyme will still reach the same Vmax as without the inhibitor. However, it will require a higher concentration of substrate in order to achieve this, and therefore the Km of the enzyme will be higher. So how does that reflect on the Lineweaver-Burk plot? So because your Vmax is unchanged, you will have an unchanged y-axis intercept. Because your Km increases, on the negative x-axis, the intercept starts getting closer to 0. So this is an example of competitive inhibition. So most statin drugs, an example of which is Lipitor, are competitive inhibitors. So here you have an enzyme, HMG-CoA reductase. This particular enzyme is an important enzyme in the synthesis of cholesterol. You have HMG-CoA, which is the substrate hydroxymethylglutaryl coenzyme A, which is the substrate for HMG-CoA reductase. So once you have this particular substrate bound to the enzyme, it indicates the committed step of cholesterol synthesis, which means once you have this substrate bound to the enzyme, it is going to make cholesterol no matter what. So if you want to reduce the plasma levels of cholesterol, the levels of cholesterol in your body, you have this lovastatin, which is a competitive inhibitor of HMG-CoA reductase. You will see that it is a structural analog of HMG-CoA. Structurally, it looks very similar to HMG-CoA. So now there is equal likelihood of the competitor binding to the active site as is the substrate, and that is how statin drugs reduce cholesterol synthesis. So just to review competitive inhibition on the Michaelis-Menten curve as well as the Lineweaver-Burk plot, your blue line is the line without the inhibitor. So it's a hyperbolic curve. Without inhibitor, the reaction is proceeding really fast. Now the red line is the one with competitive inhibitor. You will notice that the reaction sort of takes a little while in getting going, but now after at a higher substrate concentration where the reaction is of zero order, it's still going to proceed independently of the inhibitor. So which means if you increase substrate concentration to a point where it's outcompeting the inhibitor, the reaction will still be able to achieve the same Vmax. However, in doing so, it's going to increase its Km. So this is the Km in the presence of inhibitor. So now, because of the presence of the inhibitor, the binding of the enzyme to the substrate is taking a little longer, which is, in other words, slightly affecting the affinity of the enzyme to the substrate, resulting in an increased Km. On the Lineweaver-Burk plot, the blue line without inhibitor, red line with inhibitor doesn't really make any difference on the y-axis because the velocity Vmax remains the same, so the y-axis intercept remains the same. However, now with the competitive inhibitor, the Km is increasing. Now note that all of this is on the negative scale. This is negative 1 over Km. So now if we were to think about the negative 1 over Km without inhibitor as being 5, so let's say this intercept is negative 5. So 1 over Km is negative 1 over Km is negative 5, so Km is equal to 0.2 without inhibitor. Now with competitive inhibitor, let's say negative 1 over Km is negative 2, because now it's getting closer to the 0. So negative 1 over Km is equal to negative 2, which makes Km 0.5. So now you see how Km has gone from 0.2 without inhibitor to 0.5 in the presence of competitive inhibitor. So Km has increased, whereas Vmax has remained the same. Now what happens in non-competitive inhibitor? Now the substrate and inhibitor are occupying different sites on the enzyme. Here you have the enzyme. This is the active site of the enzyme. You have the substrate coming in and binding to the active site, forming the enzyme substrate or ES complex. And then you have the non-competitive inhibitor. The non-competitive inhibitor is binding to the enzyme on a site other than the active site. Okay. Now the thing about the non-competitive inhibitor is it can bind to the enzyme when the enzyme is all by itself, like in this case, or even after the enzyme is bound to the substrate, like in this case. So in both the cases, once the non-competitive inhibitor has bound to the enzyme, the reaction can no longer proceed. So because now the reaction can no longer proceed, an enzyme molecule has become kind of useless, it has decreased the efficiency of the overall reaction, which means now the Vmax is going to decrease. Increasing the substrate concentration is not really going to help overcome the inhibition, because no matter how much substrate you have, if you have enough of the inhibitor, it's always going to bind to the enzyme substrate complex and stop the reaction from proceeding. So what really affects here is the maximum Vmax that can be reached, which is decreased in the presence of non-competitive inhibitor. However, the affinity of the enzyme to the substrate, Km, does not change at all. Non-competitive inhibition does not really interfere with the substrate binding. How does it affect on the Lineweaver-Burk plot? You have the Vmax decreasing, so you have y-axis intercept getting away from 0, whereas the Km is unchanged, so the Km on the x-axis does not really change. An example of non-competitive inhibition is a HIV drug called AZT or Retrovirr, which binds non-competitively to the reverse transcriptase. The reverse transcriptase is the enzyme that makes more and more copies of HIV, and Retrovirr, by binding non-competitively to reverse transcriptase, reduces its Vmax. Thereby, it reduces the speed at which the HIV can make more and more copies of itself. So non-competitive inhibitor on the Michaelis-Menten and Lineweaver-Burk plot. So what's really happening here is the blue line is without the inhibitor, the reaction is proceeding well, hyperbolic. With the non-competitive inhibitor, the Vmax is decreasing. Now, what's really not changing is the affinity of the enzyme to the substrate. Okay, because the substrate can still go on and bind to the active site. The non-competitive inhibitor is not really interfering with the binding of substrate to the active site, but in spite of the Vmax getting changed, the Km remains the same, because the Vmax for the blue line is out here. That would make half the Vmax here, intercepting the x-axis out here, so that is the Km without the inhibitor. The Vmax in the presence of inhibitor is the green line, which would make the Vmax somewhere around at this point, which would make half the Vmax somewhere around this point, again bringing down Km to the same x-axis intercept. On the Lineweaver-Burk plot, the Km is unchanged, so you're still going to have the same x-axis intercept with or without the presence of non-competitive inhibitor. Now, remember that it's a reciprocal plot, so your 1 over Vmax in the presence of inhibitor is going to be above the 1 over Vmax without the inhibitor, because now if we were to look at this again in terms of numbers, so here your 1 over Vmax intercept is 2. So 1 over Vmax is 2 without inhibitor, so Vmax is 0.5. Now, let's say in the presence of non-competitive inhibitor, the intercept now is 5. So 1 over Vmax is 5, which means Vmax is 0.2. So you see how your Vmax has decreased in the presence of non-competitive inhibitor. So this is an example of what you can expect on the exam in terms of the Lineweaver-Burk problem. So just like, so you have your intercepts on the x and y axis in the presence and absence of inhibitor, and you need to find the Km, Vmax, and find out what the order of the reaction will be. So I'll leave it for you guys to work this problem out at home, and then we can discuss this in the class. So what are the different factors that regulate the enzymatic activity? Number one is the substrate concentration. So if you have an increase in substrate concentration, that increases the enzymatic activity until the substrate concentration returns to normal. An example of which is an enzyme called alcohol dehydrogenase. Alcohol dehydrogenase is an enzyme that converts alcohol into acetaldehyde, which is even more toxic than alcohol itself. But then that acetaldehyde is quickly converted into acetate, and other molecules that can be utilized by the cells. So if you ever had an evening out drinking alcohol, you would expect an increase in the enzymatic activity of alcohol dehydrogenase right after the consumption of alcohol. That is because this particular enzyme is now being called into action to uh, convert the alcohol into acetaldehyde and then further convert that into acetate. So increase in substrate concentration increases the enzymatic activity, which alcohol dehydrogenase on other occasions might not be that active as an enzyme. The second factor is product concentration. A lot of metabolic processes in our body have feedback loops. As soon as the product of a reaction accumulates, it can inhibit the enzymatic activity. A lot of these enzymes are making nucleotides and so on. So as soon as you have enough of the nucleotides, the enzyme has to stop making any more of them because that would be redundant. A case in example is an enzyme called 5-PRPP, 5-phosphoribosyl pyrophosphate. You will learn more about this in the chapter called Nucleotide Metabolism, but 5-PRPP is one of the important enzymes that is involved in the committed step of nucleotide synthesis. So as soon as you have enough of the nucleotides, you have inhibition of this enzyme so as to not make any more nucleotides. Enzymatic activity can be regulated by some allosteric binding sites. In biochemistry, allosteric regulation is the regulation of a protein by binding of an effector molecule at a site other than the protein's active site. Okay, the site to which the effector molecule binds is termed the allosteric site. Allosteric sites allow effector molecules to bind to the protein, often resulting in a structural change that can affect protein dynamics. Effectors that enhance the protein's activity are referred to as allosteric activators, whereas those that decrease the protein's activity are called allosteric inhibitors. An example of which is hemoglobin. Now, hemoglobin binds to oxygen. There are 4 subunits of hemoglobin that bind to oxygen. However, once you have the first subunit bind to the oxygen, it increases the affinity of other 3 subunits to oxygen. So in this case, you have a positive effector or oxygen acting as an allosteric activator. And then you have negative effectors or allosteric inhibitors that inhibit enzymatic activity. You have 2,3-bisphosphoglyceric acid or 2,3-BPG, which is present in human red blood cells. 2,3-BPG binds with greater affinity to deoxygenated hemoglobin, that is when the red blood cell is near the respiring tissue, than it does to oxygenated hemoglobin. So 2,3-BPG interacts with deoxygenated hemoglobin by decreasing their affinity for oxygen. So it allosterically promotes the release of remaining oxygen molecules bind to hemoglobin, thereby enhancing the ability of red blood cells to release oxygen near the tissues that need it the most. Now there can be 2 different kinds of allosteric regulators. One where the substrate of the enzyme itself is an effector, in which case it is known as homotropic effector, or it can be a molecule that is different from the substrate, in which case it is known as the heterotropic effector. So in coming back to homotropic effector, the substrate itself is the effector. An example is binding of one molecule of oxygen to one subunit of hemoglobin increases the binding of oxygen to other subunits. Oxygen is the substrate for hemoglobin, and binding of first oxygen molecule increases the binding of other oxygen molecules, making oxygen a homotropic effector. Enzymes that have homotropic effectors usually show the principle of cooperativity, and therefore they exhibit the sigmoidal curve. In heterotropic effector, the effector itself is different from the substrate. Typically enzymes that have heterotropic effectors follow feedback inhibition. If you notice in this particular schematic here, if you consider this to be a particular metabolic pathway, you have G, which is not the substrate for this particular reaction, which is actually inhibiting this reaction, the particular enzyme carrying out this reaction. So G is the homotropic effector of the enzyme that's converting D to E. An example is phosphofructokinase. PFK1 is an enzyme that catalyzes the most important step of glycolysis. It converts fructose 6-phosphate into fructose 1,6-bisphosphate, which is the committed step of glycolysis. Once this reaction has completed, it is going to end up making more glucose. So what inhibits PFK1 is citrate. Citrate is not a direct end product of this reaction and is therefore a heterotropic effector of the enzyme phosphofructokinase 1. You will learn more about this, by the way, in glycolysis. Another way to regulate enzyme activity is through covalent modification. The most common form of covalent modification is phosphorylation or dephosphorylation by the addition or removal of phosphate groups from the serine, threonine, or tyrosine residues of the enzyme. So here you have protein kinase, which is an enzyme involved in phosphorylation. Protein kinase uses a phosphate group from ATP, which in this case is the phosphate donor, and it attaches that phosphate group onto an enzyme, in turn phosphorylating this enzyme. Another enzyme, which is phosphoprotein phosphatase, can then dephosphorylate this enzyme. It can remove this phosphate group and convert it back into its original form. So depending on the enzyme, the phosphorylated form may be switched on or it can be switched off. For the most part, phosphorylation of an enzyme makes it active, but there can be some examples where phosphorylation actually switches the enzyme off. An example of which is glycogen phosphorylase. Phosphorylation of glycogen phosphorylase increases its activity, switches it on, whereas phosphorylation of glycogen synthase switches it off or decreases its activity. And then the last method of regulation of enzymatic activity is induction and repression of enzyme synthesis. Cells can regulate the amount of enzyme present by altering the rate of enzyme synthesis. These enzymes are not needed to be present in the body at all times and are often needed only at a particular stage of development or under selected physiological condition. An example of which that we talked about some time back was alcohol dehydrogenase. Your body is going to make more of alcohol dehydrogenase when it needs to metabolize alcohol. Now, regulation through this method of induction and repression of enzyme synthesis takes a long time to take effect, whereas regulation by allosteric regulators is almost instant. So another example is elevated insulin levels resulting from high blood glucose increase the synthesis of enzymes involved in glucose metabolism. Your body is going to make more of the enzymes involved in glucose metabolism if there is a lot of glucose present. In other times, it is not going to make more of those enzymes. So from a clinical perspective, enzymes are extremely important as a part of diagnosis. Most enzymes are present in cells at much higher concentrations than in blood plasma. Though many enzymes have no function in plasma, they are still useful as diagnostic tools. Measurement of their levels in plasma offers valuable information about diseases involving the tissue of their origin. It is much easier to measure enzyme activity in the body fluids by monitoring changes in either substrate or product concentrations than to measure enzyme protein concentration. The enzyme activity in plasma may be increased due to proliferation of cells, cellular damage, increased enzyme synthesis or because of reduced clearance from plasma. The enzyme activity in plasma may be lower than normal, very occasionally due to decreased synthesis or some congenital deficiency. Changes in plasma enzyme activities may sometimes help to detect and localize tissue cell damage or proliferation, or to monitor treatment and progress of disease. Elevation of certain enzymes in plasma is indicative of tissue damage. During tissue damage, the enzymes within the intracellular fluid spill out into the plasma. An example is alanine transferase ALT levels. If you ever had a metabolic panel done, your ALT levels are measured, which are indicative of the health of your liver. High ALT levels are indicative of hepatic damage. Similarly, creatine kinase and lactate dehydrogenase, both of these enzymes are found in abundance in heart tissue, and any increase in plasma levels of creatine kinase and lactate dehydrogenase is indicative of myocardial infarction. And that is it about this long chapter, enzymes.